Our hands, our eyes, our face, our whole body is a swarm of billions of cells. At the heart of each of them nestles the DNA we received from our parents and pass on to our children.
What exactly is transmitted from one generation to the next, from one cell to the next? We know that DNA is transmitted, but not only DNA.
Identical twins have the same DNA, the same genome, yet they're physically different. How is this possible?
I think if we view the genome as a book, then it can be read in many different ways.
Scientists have long thought that DNA alone ruled our biological destiny. Today, they're discovering another world.
We've become more and more aware that DNA doesn't explain everything, and more capable of integrating the ideas about the impact of the environment.
All over the world, biologists are exploring this new mystery.
My work consists in looking at the heart of cells of new knowledge and going where no one has been before.
A scientific adventure where the researchers are gradually discovering what influences our DNA throughout our lives, our identity, and perhaps our heredity. In the natural world, one insect has always fascinated: the bee. Its destiny never ceases to amaze and puzzle the specialists. They are gradually elucidating the mysteries of its development. There are hundreds of species of bees in existence, including this one, the domestic bee, which lives in a highly organized society.
I'm standing in front of a colony of bees. A colony of bees means a large number of workers, maybe 20, 30, or 40,000. And there's just one queen, the mother of all those bees. You can see the queen here. She has a dot of red paint on the thorax, put there by the beekeeper to help him find her. Though she's quite clearly different from the other bees around her. She doesn't have the same shape. She's longer. You can also see that her thorax is slightly larger. The queen's distinctive feature is fertility. She's the only one able to mate, so her job is to lay eggs.
A queen is therefore very different from the other workers in her colony. And yet, in the very first days of their lives, the queen's larvae and the workers' larvae are identical. How is it that occasionally one of them steps from the ranks? It wasn't until the 1950s that this mysterious process was understood.
What makes one larva a queen rather than a worker is largely explained by what it eats. A larva eats royal jelly for 3 days, and then, if it is destined to become a worker, its food will be changed and the royal jelly will progressively be mixed with larval mush that contains honey and pollen. The queen larva, however, will continue to eat royal jelly for the rest of its time as a larva.
Thus, a simple change in diet can cause profound differences between two living beings that were so similar when they started out their lives. But how can you explain that an external element can have such an impact on their growth? Isn't that supposed to be governed by their DNA? To understand this process, you have to go back to the origins of one of biology's most fascinating scientific adventures, the sequencing of the human genome. We have long sought to understand the laws of heredity and the way character is transmitted from one generation to another. In the mid-20th century, we discovered that DNA was the medium of heredity. In humans, at the heart of the cell, the nucleus, are 23 pairs of chromosomes. Each chromosome is made up of 2 strands of DNA. Composed of 4 bases, represented by the letters A, T, G, C, the DNA of a human being contains 3 billion letters, which assemble themselves into phrases to form genes. The genes are used to make the proteins, which make up the building blocks of living organisms. In the early 2000s, the sequencing of the human genome, which draws a detailed map of over 25,000 genes, was completed. It was a significant scientific breakthrough, and hopes were high.
Messieurs, bonsoir. Voici les titres de l'actualité de ce lundi. Des généticiens de 18 pays sont parvenus à dresser une carte presque complète du génome humain. C'est une percée scientifique capitale.
Today, we celebrate the revelation of the first draft of the Human Book of Life.
Scientific journals published the genome map, with pride of place being given to the almighty gene. Genome sequencing raised the wildest expectations. Genes for obesity and schizophrenia would be discovered. Cancer would be eradicated. The promises of science seemed endless.
We are probably, as scientists, a little bit to blame that we told everybody how important DNA was.
We said we had deciphered the human genome. Decipher means taking an encrypted message and making it intelligible. But we read the genome and understood nothing. The actual deciphering is what we do afterwards, what we've not finished doing, and it'll take some time.
The scientists know they're only at the start of the adventure. They have in their hands a text that is difficult to interpret.
DNA is not the only critical thing. We cannot explain everything just by looking at DNA sequence. We needed to know what the sequence is, but biological complexity and biological systems are not just about DNA. They're about the way DNA is used, the way DNA is read, the way DNA is transmitted.
A few months after the announcement of the sequencing of the human genome, the review Science for the first time devoted an entire issue to epigenetics, a new scientific field that explores what influences the expression of DNA. For there are too many questions unanswered.
People were searching for something else that might explain the beauty, the complexity of who we are.
In Australia, researchers are striving to understand the fate of bees. Their genome has just been entirely sequenced. And there's a big surprise. There is no genetic difference between the larvae of a worker bee and the larvae of a queen. They share the same DNA. So if the difference between larvae is not genetic, perhaps it's epigenetic. Might this famous royal jelly be able to influence bee larvae genes to the point where it can turn them into a queen? The process begins at the larval stage. After 3 days, worker larvae and queen larvae are no longer fed in the same way. It's a change that profoundly alters their development.
Here, we see that they metamorphose at different rates. The queens grow faster than the workers. After around 2 weeks here, a queen is ready to emerge, whereas a worker will take 3 weeks.
So it is the feed, the royal jelly, which makes the difference. But how does it act on the larva genes?
In chemical terms, what happens is an epigenetic mechanism, the methylation of the DNA which triggers these various growth programs.
bee DNA contains around 10,000 genes. As with all living organisms, some genes are active, or expressed as they say. Others are inactive. The expression of genes can be inactivated by what is called the methylation of the DNA. It's a simple chemical mark that attaches itself to the gene and in a way enables it to be turned off like a switch.
If there's a lot of DNA methylation during early growth, then you'll get workers. Whereas if the DNA methylation is turned off, then you get queens. In the same way that food activates a certain epigenetic program that enables either queens or workers to be created, here we're injecting molecules into these eggs to stop DNA methylation. And so, all these eggs will grow into queens.
Thus is the amazing recipe for making queens revealed to us. There is no queen DNA, nor worker DNA, just chemical modifications around their genes which play a key role in their growth. This applies to bees. Does it also apply to other species, other queens? Jonathan Weitzman has his own ideas on the matter.
Another example from nature. I'm British. To the left is our queen by birth, so we can say she's a genetic queen. To the right are the bees, who have just one queen, but she's not born to be queen. In time, one of the bees becomes a queen because she eats royal jelly. So you can see, in England, there are two ways of becoming a queen. Either genetically, on the left, or by interaction with your environment. You can, in a few rare cases, become a queen.
What are we made of? Who are we? Whether it's a matter of scientists or cells, DNA doesn't explain all the diversity of life. Cells from our liver, our eye, or our hand all have the same genome, and yet they're almost as varied as the researchers. So how do you explain such diversity? This question fascinates Jonathan Weitzman. He explores the issue of identity, starting with that of our cells.
So in our body, we have hundreds of different cell types, cells that behave differently, that have different characteristics, and they transmit those different characteristics to their daughter cells. The hepatocytes in the liver, the neurons in the brain, the lymphocytes in the blood, the keratinocytes in the skin. All these cells behave differently. They use the genome differently. And yet, they all come from the same original cell, the fertilized egg. How did the first cell give all this diversity of cellular states with the same genome?
It is a question that Scottish biologist Conrad Waddington began to answer in the 1940s. He was the first to coin the term epigenetic. Fascinated by the growth of organisms, Waddington wondered how an embryo became a being made up of many varied cells that all sprang from a single cell.
Conrad Waddington drew a picture of a landscape, of a mountain, and at the top of the mountain he placed a cell or a bead. And the idea was that at the top of the mountain, the cell could become many different things, depending on which path down the mountain it took. So at the top of the mountain is like the multipotent stem cell that can become any different type of cell. And as the cell moves down the mountain, it becomes more and more committed to a particular path, depending on which valley it goes into, and that will determine its fate.
Liver cells, heart cells, skin cells, brain cells. The fate and the identity of our cells are not only etched in our DNA. Other mechanisms come into play so that cells differentiate, and more especially, that they keep the memory of their identity.
We all know that we began as an egg. From that point, each being develops with this epigenetic function that enables genetic data to be read. There's something a bit magic in all that. It's wonderful to think that we're trying to understand how it works, how I or how this plant are made from this genetic data, which has been changing for 2 billion years. And then these epigenetics can read the system and make the plant or the human being that I'm looking at.
The genome will not change during the differentiation process. What will change is the way the genome is used. And that's epigenetic mechanisms that will control that will determine which parts of the genome are used and which parts of the genome are not used.
The way our cells are expressed is therefore critical to our growth and that of our cells.
There have been many ways to think about epigenetics. One metaphor that does work well is music. Music is written with lines and dots, and yet the lines and dots are critical to playing the music. But every time those lines and dots are played by a musician or by an orchestra, they will sound different. So the music written down could be like the DNA, and the expression of that requires a musician and an interpretation.
The same genetic partition can have several interpretations. Who better to understand this than twins? Born from the same egg, identical twins share the same DNA. Are there differences of interpretation in their genome?
I love garlic.
I hate garlic.
He runs much faster than I do.
He needs to practice more than I do.
My French isn't bad.
I don't speak French.
Jonathan Weitzman and his twin Matthew are both researchers. They share the same DNA and the same questions about identity.
Yeah, maybe we're both interested in genetics because we've always been interested in what's transmitted, what makes us who we are.
We're a combination of our genome, our epigenome, our experiences. That's what makes me who I am today. But those experiences and the changes in our epigenome will tell me who I am tomorrow.
The genome is relatively static. The epigenome is, in comparison, relatively dynamic. Identity, who we are, is something that changes all the time throughout life. And so the epigenome must in some way carry memories of that past and probably contribute to defining who we are. So the longer we live, the more our epigenomes will diverge. And there are studies that have shown, or begun to show, that this is the case.
So our genes are being influenced throughout our lives. Our experiences have an impact on their expression. How is this expressed in two people with the same DNA, like identical twins? That's what Manel Esteller set out to find here in Barcelona.
¿Cómo estamos?
Bien, hola.
Berta.
Sí, Grisela.
Bueno, bienvenidas. Como sabéis, estamos estudiando nosotros la epigenética, alteraciones o cambios que podéis tener en vuestro material genético a pesar de ser gemelas. Son cambios normales, no hay nada que preocuparse. Puedo explicar algunas diferencias que tengáis entre vosotras. ¿Diréis que ahora sois más iguales o más distintas que hace 10 años? ¿Os vais separando poco o vosotras todavía sois de las muy cercanas?
Y físicamente yo creo que nos vamos diferenciando un poco. Con los rasgos van cambiando.
Sí, sí. Lo que sí, los gemelos monocigóticos también podéis ser distintos, serían las huellas dactilares, ¿no? Cuando os miráis las huellas de los dedos, ¿las veis distintas? Este es un cambio puramente epigenético, sin ninguna alteración genética, que sucede pues, en los dedos aquí, ¿no? Para diferenciar.
Se parecen mucho, pero se pueden
Hay
distinguir.
ciertas diferencias en las mismas.
Sí.
Esto es de aparición en el nacimiento, al nacer
Ya
ya estaba.
nací con esta mancha y ella pues nada. ¿Este cambio a qué se debe?
Este cambio no lo sabemos. Podría ser que hubiera una pequeña mutación, un cambio genético, o puede ser un cambio epigenético asociado a las mismas. Esto, hasta que se mire a detalle, no lo sabremos. Lo que sí podemos decir es de que si sois muy parecidas, es genéticamente, sois idénticas, pero epigenéticamente estáis divergiendo. Estás divergiendo porque los estilos de vida van siendo distintos, ya se ve físicamente y también en el crecimiento que tendréis y también en las enfermedades. Eso va a cambiar y va a ser muy importante para ser dos individuos que no son fotocopias. Las conclusiones derivadas del estudio.
Studies on twins show that the DNA conditions what we are and the diseases we develop, but it's not a closed book.
Pero no es un libro cerrado.
These studies are essential in demonstrating that there is a certain genetic determinism, but not 100%.
Studies on identical twins is a way to better assess the influence of epigenetics on the development of individuals. But do they also play a role in the appearance of illnesses?
Studies on identical twins are also important in the field of cancer research. With sisters who have inherited a genetic mutation that gives them a higher risk, let's say 80%, 90%, of contracting breast cancer, it can happen that one of the sisters develops cancer at 60 and the other never, or at 90. How is this possible if the DNA is the same? Well, it's because there are epigenetic differences.
So there are epigenetic differences between twins, differences which are sometimes the sign of anomalies and could be used as flags in checking for the appearance of illnesses like cancer. By exploring these new territories, teams like that of Edith Hurd at the Institut Curie bring renewed hope to this field.
We know that cancer is a genetic disease. We know that changes at the DNA sequence level are important in the cancer process. However, we're starting to realize more and more that it's also an epigenetic disease, that there are changes in gene expression that don't necessarily involve a change in DNA sequence that are actually implicated in the progression of tumors.
Edith Hurd's team has just broken new ground in understanding these mechanisms. The researchers focused particularly on a fascinating chromosome in female mammals, the X chromosome. In human beings and in most mammals, the sex is determined by the X and Y chromosomes. Females inherit 2 X's and males 1 X and 1 Y. But with 100 genes on the Y against 1,300 on the X, there's a clear imbalance. To restore the balance, one of the 2 X chromosomes is inactivated in females right at the start of their embryonic development. In fact, it's an epigenetic mechanism which contributes to silencing one of the X chromosomes and, above all, of maintaining that silence throughout a female's life.
X inactivation is essential. If one of the 2 X chromosomes is not shut down during development of a female, the embryo dies very early on. This illustrates how important it is to get gene dosage right. If you have an excess of expression of genes from the X chromosome, that leads to lethality.
Feline or human being, this process is in fact inevitable in most female mammals. To Edith Hurd, this vital phenomenon provides a lot of information on how normal and cancerous cells function. Throughout our existence, our cells are renewed by division. And with each division, epigenetic mechanisms keep the memory of the X in activation. But what happens when the cell loses that memory? That's what researcher Ronan Chalinier is trying to find out. In their normal state, in the nucleus of these cells, the inactive X is very inhibited, as in this diagram. But in the breast cancer, its aspect changes.
In tumor cells, we've discovered in recent years that the inactive X was still present, but there was a change in its silent state. This inactive X has a far more relaxed, less dense structure. We've noticed that in cancer cells where the epigenetic mechanisms are disturbed and deregulated, there's a reactivation of certain inactive X genes which are normally suppressed.
But the fact that some genes are reactivated favors the growth of tumors. This research opens new avenues of action against cancer.
Epigenetic changes also provide hope in cancer because epigenetic changes can be reversed. Unlike DNA sequence mutations that can't really be reversed in any easy way, this contrasts with an epigenetic change which can indeed be reprogrammed or reversed with certain molecules. So currently there's great hope that some types of cancer could actually be treated by drugs that target the epigenetic machinery.
Molecules which target these processes, epidrugs, are currently undergoing trials. But researchers have discovered that medication marketed several years ago unwittingly affect epigenetic mechanisms.
In fact, one drug that has been used for many, many decades turns out to be an epidrug. Decitabine, as it's called, is used to treat myelodysplastic syndrome, which is actually a blood disorder that leads eventually to leukemia. And so it was discovered many, many years ago as a successful treatment in slowing down the progression of this disease. So this is an example of an epidrug that is used in the clinic that is successful. What's important to know, though, is we don't actually understand the targets of these epidrugs. We know that some of them can work in the clinic, but the challenge now is to understand how they do it.
Epidrugs present a promising lead in the fight against cancer. The role of epigenetics in the development of individuals and of certain diseases is gradually becoming clearer. But in the same way that genes are passed from one generation to another, could these mechanisms that influence the gene expression also be transmissible?
What type of information is transmitted to the next generation is still a very open question. We know that characteristics have been transmitted. I look like my father and my mother. My son and my daughter look like me. So where does this come from? We focused a lot on the DNA, mainly because we could. It's easy to measure. It's easy to find the sequence. Now we're interested more and more in what's transmitted with the DNA, or around the DNA, or above the DNA.
How can we get a better insight into this transmission? Currently, the best clues are supplied by plants. Geneticist Vincent Collot has carried out research on a little plant from the same family as mustard, a plant called Arabidopsis, or mouse-ear cress.
Arabidopsis is a plant which is of great interest to geneticists because it is very prolific. It has a very short development time, in other words, it can go from seed to seed in less than 2 months, and the cherry on the cake, we know it has a very compact genome.
Can Arabidopsis pass on to subsequent generations visible changes such as longer or shorter roots, or earlier or later flowering without this being written in the DNA? To find out, researchers induced epigenetic modifications in an Arabidopsis plant. They then crossed this plant with a wild plant. And after successive cross-breeding, they obtained a family of plants over several generations. The first major discovery: these epigenetic modifications are transmitted and remain stable through at least 16 generations. The second observation: these modifications are associated with visible changes, such as root length or the flowering period. So, for the first time, scientists are able to show evidence that characteristics can be transmitted over a large number of generations without any change in the DNA sequencing.
What we also found was that this epigenetic variation passed on through the generations has a lower stability than that of the states of DNA sequencing. The sequence of the DNA is ATGC, written in a particular way and transmitted with extreme fidelity, unlike the epigenetic states, which evidently appear to be less stable. In other words, this transmission we're measuring will last for a certain number of generations, tens or even hundreds of generations, but certainly not millions of generations.
Thanks to this experiment, scientists have evidence of the transmission of epigenetic marks, which modify certain aspects of the plant. In this case, the modifications have been induced in a first generation of plants in the laboratory. But what happens in nature? Can these changes be induced by the environment, in case of drought, for example?
We're facing an unknown, the part environment plays over the way genes function. But how far does that environment dictate heritable changes over generations? That's an open question. Does environment play a part?
To answer this question, Vincent Coulot and his researchers at INRA have launched a new experiment. They have installed hundreds of genetically identical plants on the conveyor belts of this system that's unique in the world, the phenoscope. The mechanical rotation system exposes the Arabidopsis plants to the same light and enables only the watering to be varied to measure its impact.
The question we're trying to answer now is, what are the conditions that lead to the appearance of epigenetic variations? Is the environment itself capable of inducing this type of change? And if so, are the changes we observe as stable as those we've managed to establish experimentally in the laboratory? Over how many generations? And with what range? That's more or less the range of questions we can tackle thanks to this totally unique experimental system, the phenoscope.
The results of this research are eagerly awaited. They will enable us to see whether environmental factors such as drought can influence the plant genome over several generations. What do we know of other species? What can we say about the impact of environment on our own genome?
It's an ongoing search and debate to understand exactly what is transmitted to the next generation, how much other information is transmitted. Do we transmit information about the environment in which we live? Some people think it may be quite subtle, fine-tuning. Some people think that it may have a very big impact.
thousands of kilometers away in the American Northwest, a researcher is convinced that environment plays a major role in our organism and that it leaves lasting traces transmissible via epigenetic mechanisms. A specialist in reproductive biology, Michael Skinner came to epigenetics rather by accident. An error in manipulation enabled him to study the effects of pesticides on several generations of rats.
Most scientific observations are not necessarily planned. And so this is what you would call a serendipitous observation. So we were studying the effects of this fungicide on the gestating female. And we wanted to see what would happen if a fetus was exposed to its health when it was born and grew up to become an adult.
The American researchers' study focuses on the effects of pesticides. They were looking to gauge their impact on the fertility of male rats. They therefore injected pesticide into pregnant females. When the babies were born, there were no anomalies. But as adults, the male's sperm count signaled a drop in fertility. These males reproduced for the first time. Their offspring, in turn, presented defective sperm. The crossbreeding continued through to their great-grandchildren. Then came a surprise. The same anomalies were observed in the male descendants, though there was no more direct exposure to pesticides, neither via injection nor via the maternal uterus. Moreover, with each generation, epigenetic modifications were discovered and correlated with sperm anomalies.
90% of the males had this reproductive abnormality that was carried for 4 generations. And so that showed us that we had a phenomena that was not following classic genetics. It was definitely different. And so we followed that up to show that there was this epigenetic transgenerational inheritance.
Have these results been confirmed? Not really, but no one has reproduced the study in the same conditions. Michael Skinner claims to have shown that pesticides can cause non-genetic anomalies that are transmissible in rodents. Some decry the work as too partial, with hasty conclusions that undermine the concepts of heredity and trigger debate.
If you're not doing something controversial, you're not doing something really important. And not that, the subtle changes are not important. It's a matter of you're not going to make the big steps in science. So if all of scientists started thinking that way, then there would be challenges to all of our dogmas much, much more rigorously than there is today. But instead, we have this tendency to accept a dogma and work within it without challenging it. So that's not the best way science should be done, in my mind.
Here and there, experiments similar to that of Michael Skinner were conducted. Here in Zurich, Isabel Mansui's team was also seeking proof that epigenetic heritage was linked to the environment. But this time the subject was stress. Is it biologically transmissible?
We used a mouse as an animal model to test the theory that exposure to traumatic stress during childhood can alter the epigenetic mechanisms permanently and modify adult behavior.
To create trauma in suckling mice, the young were separated from their mothers unexpectedly and repeatedly.
What we were able to show is that exposure to periods of chronic stress altered the mouse's behavior and also, in parallel, altered a certain number of epigenetic mechanisms in the brain and in offspring, and that these epigenetic modifications were passed through the generations.
The baby mice that grew up in these conditions were depressed as adults. They evaluated danger less well and took more risks. Disorders that they passed on to their descendants as far as their great-grandchildren.
An important lesson we can draw from these results is that not everything resides in the genes, but the environmental factors to which we are exposed, in particular trauma during childhood, are very important and can determine our behavior and our life across several generations. What excites me about the field of epigenetics is the consequences, the applications for human beings, especially in psychiatry, because there are many psychiatric illnesses like depression, schizophrenia, personality disorders that aren't known. We don't know the causes, we don't know the mechanisms, and we think that our research into epigenetics could lead to a better understanding of these diseases.
It is much too early to say whether we can pass on stress as the mouse could. According to the work of Isabelle Mansuy and Michael Skinner, genomes are like sponges, absorbing the slightest effects of the environment. Effects that leave traces that are passed on for several generations. These conclusions are the subject of debate within the scientific community.
In fact, if we think about it, if we were that susceptible to environmentally induced epigenetic changes, we'd be a mess. Our cell types would change identity, probably tumors, and actually we'd probably not even be here. We wouldn't be alive.
How far are we affected by our environment, and what do we pass on to our children? In reality, the influence of epigenetics comes up against limits that have just been discovered. In sperm and ova, then at the moment of fertilization, a large portion of the epigenetic marks are erased, enabling a kind of reboot for the new generation. But this spring cleaning is incomplete. Some marks remain in place. Which ones and why? That's the mystery that Wolf Reich and his team are working to solve in Cambridge.
One aspect of the transgenerational epigenetics is probably incomplete erasure. We would like to understand what's going on. The choice between what's erased and what is not erased. And also if that choice could be changed. So for example, if the environmental conditions are different, if the food supply is different, if the nutrition is different, is there a mechanism by which you could switch this choice between erasing information and giving it to future generations? It is a very, very exciting question.
This selective erasing can be seen under the microscope. These are very early embryo cells. The nuclei are blue, with practically no epigenetic marks. In purple are the few marks retained that were transmitted by the parents. But take a look a few days later. The cells, now more developed, at a more advanced embryonic stage, are already harboring a constellation of new epigenetic marks linked to their growth.
Probably for the last 10 years or so, we have been very actively studying this erasure process because it really fascinated us. I think there are many unexplored areas. It's probably true that the epigenome is influenced by nutrition, by the food that we have, by our, where we grow up and environment and things like that, and also potentially what happened to our parents in terms of the environment, the food and things like that.
A few paces from Wolf Reich's laboratory, still in Cambridge, a team of researchers wanted to clarify another issue. Is food capable of marking our DNA? And if so, do we pass this on to our descendants? Anne Ferguson Smith is examining the impact of food deprivation on a pregnant mouse, on her young, and on subsequent generations.
When a mother is calorically restricted during pregnancy, this can have quite profound effects on her offspring. In this particular mouse model, it's a 50% caloric restriction. That's a very severe undernourishment. And the mice are born small, the babies are very small, and they go on in adulthood to get fat and diabetic. They don't metabolize glucose properly or insulin properly, and they have diseases very similar to what we see in our population today with this increased incidence of adiposity, obesity and diabetes. Because in this model, if you continue to undernourish the embryo after birth, the symptoms don't arise and actually the animals are very healthy.
It's as if the baby mice exposed to food deprivation in the womb were programmed to adapt to this shortage. And when they grow up in an environment where food is abundant, they are no longer well adapted and develop diabetes and obesity. The next generation, the grandchildren, show the same symptoms. How many generations can be affected by these illnesses? The research on mice is continuing. But how can we find out whether food is capable of having such an influence in humans?
So obviously one can't do experiments on human populations like this, but we do know that in history there are situations where there have been similar environmental compromises to pregnant women, in particular the Dutch hunger winter of the Second World War.
During the winter of 1944, part of the Netherlands suffered greatly from famine. In order to punish them for their support of the Allies, the Nazi occupants blocked all food deliveries. The survivors and their descendants are of particular interest to researchers in epigenetics.
So these are very moving pictures, and in fact here there are pregnant women, and you can really see the challenges that they must be facing in this time of deprivation. The children of these pregnant women have been analysed and have been shown to have increased incidence of adult onset diseases such as diabetes, obesity, neurological disorders such as anxiety and depression and schizophrenia, and also an increased incidence of cardiovascular disease. But what has been discovered is that the offspring of the sons of these women are heavier as adults. So this does tell us that the environment is having some kind of effect over at least a couple of generations.
Research devoted to the impact of the environment on the genome has only just begun, and there are a number of mysteries to be cleared up.
So genetics is wonderful. It's the most important mechanism for transmitting healthy and unhealthy outcomes from one generation to the other. But what is emerging now is that there's something else on top of the genetics that can have an impact on health and well-being from one generation to the next. And to me, it's an extremely exciting prospect trying to understand what that mechanism might be, that non-genetic mechanism for conferring this intergenerational effect.
What excites me about epigenetics is that it gives us lots of ways to express a static genome, and it gives us a way to look at the traces of environmental impacts and the traces of our own personal histories on the way genomes behave. And I think what I like particularly is that it's reversible. It's sort of optimistic that nothing is set in stone.
Effectively, this widens the field of genetics. We're not dealing with something that is passed on unchangingly, but there's a wealth of possibilities between an almost evanescent transmission to something that is virtually engraved in marble. And epigenetics occupies a large part of what isn't written in stone.
Epigenetics is now in full swing. Research is growing all the time. But it will take a few more years and a good many studies to fully grasp the subtleties of life's lush musical score.